Hey guys, in this video we're looking at cell biology focusing on microscopy. So microscopes let us see things that we can't see with our plain eyes. Microscopy techniques we can use have developed over the years as technology and knowledge has improved. And here we can compare light microscopes with electron microscopes. So the first thing is that light microscopes use light and lenses to form an image of a specimen and magnify it, which means they make it look bigger. They let us see individual cells and large subcellular structures, for example, nucleus. Electron microscopes use electrons instead of light to form an image. They have a much higher magnification than light microscopes. Microscopes also have a higher resolution, which means it has a higher ability to distinguish between 2 points. A higher resolution gives us a sharper image. And lastly, electron microscopes let us see much smaller things in more detail, for example, a mitochondria. So this is a magnification formula, which is magnification equals image size divided by real size. If you're trying to work out the magnification or the real size or the image size, you cover up what you're trying to work out and then you have the formula. So here's an example question. A specimen is 50 nanometers wide. Calculate the width of the image of the specimen under a magnification of times 100. Give your answer in millimeters. So what you will have to do is use the formula image size equals magnification times real size. So you'll do 100 times 50 to get 5,000 nanometers and then compare that to millimeters, so you'd get 5 millimeters. Standard form. So microscopes can see tiny objects, therefore it is useful to write numbers in standard form. This is where you can change very small or very big numbers with lots of zeros into something more manageable. For example, 0.017 can be written as 1.7 times 10 to the power of minus 2. To do this, you just need to move the decimal point left or right. The number of places the decimal point moves is then represented by a power of 10. This is positive if the decimal point has moved to the left and negative if it's moved to the right. Standard form example then. So a mitochondrion is approximately 0.0025 millimeters long. Write this figure in standard form. The first number needs to be between 1 and 10, so decimal point needs to move after the 2 here. Count how many places the decimal point has moved. This is the power of 10. Don't forget the minus sign as the decimal point has moved right. So 0.0025 can be rewritten as 2.5 times 10 to the power of minus 3.